This is episode 317 of the Stem Cell Podcast, Engineering the Human Heart, with Dr. Aitor Aguirre. Hello, everybody. This is Dr. Dalen James with Dr. Arun Sharma. Welcome back to the Stem Cell Podcast, where we culture knowledge in stem cell research by talking to some of the brightest minds in the field. The pod is brought to you by STEMCELL Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. If you're enjoying the listen, rate us, leave a review. We're always looking for feedback on how the podcast can be improved and for suggestions on the guests we should have on the show. Today, we have Dr. Aitor Aguirre, who's from Michigan State University. He's on the podcast to talk about his research understanding human heart development and disease using stem cells and cutting-edge organoid tech. We've also got our usual roundup of recent highlights and stem cell news that's coming up in just a sec.
But first, join the Global Stem Cell event, the ISSCR 2026 annual meeting from July 8th to 11th in Montreal, Canada. Connect with nearly 3,500 scientists, clinicians, and industry leaders for 4 days at the frontiers of stem cell science, regenerative medicine, and engineering biology. From fundamental discovery to clinical breakthroughs, this is where collaborations start and new ideas take shape. Be part of the global conversation shaping the future of stem cell research and submit your late-breaking abstract by April 22nd. Visit isscr2026.org, where you can sort the agenda by theme. Topics include bioengineering, organoids, neural stem cells, aging, and more. Speaking of organoids, we're going to start things off with an organoid paper. A little bit different here. It's more on the topic of using organoids as an infectivity model. Title here is a human cerebral organoid model of West Nile virus encephalitis shows innate immunocompetency. And we've covered different versions of infectivity organoid models in years past, obviously, because there was COVID. But even before that, Zika, I don't know if you remember, we were covering a lot of Zika organoid papers in like 2019 before the onset of COVID. But this is in that same kind of wheelhouse, looking at different infectious pathogens and how they would be able to influence viral tropism, in particular tropism towards brain tissue, right? Which is, of course, hard to obtain, which is why they used the cerebral organoid model here. So West Nile virus, I remember first hearing about West Nile virus, and there's a breakout or something back when I was in elementary school. I remember folks freaking out about it and be like, oh yeah, be careful of the mosquitoes. They can really spread West Nile virus. And, we had to stay inside for a bit more over the summer. I remember in Alabama where it was hot and humid, lots of mosquitoes over there. So West Nile virus has been around for a while. I mean, it's an arbovirus of particular global interest. And I feel like we have these ebbs and flows of the different, not pandemics, but different epidemics of West Nile virus infections over the years. This is particularly nasty because it can cause neuroinvasive disease in humans. So there's no protective vaccine or specific treatment available for human West Nile virus encephalitis, which can arise from West Nile virus infection. Now, the virus can cause pretty serious situations here, kind of analogous to what you might see in Zika, but of course in more of an adult state. The virus can cause neuronal cell death, while astrocytes and microglia cells are suspected to contribute to the West Nile virus pathology. So there's perhaps a cell-cell crosstalk happening there. And because of that potential cell-cell crosstalk, you got to figure out the mechanisms of the infection here. So understanding the role of the different cell types in the infection processes is critical. And critical for developing potential future therapies too. So here, this is a study coming from the lab of Dennis Tapp from Hamburg, Germany, the Bernard Nocht Institute for Tropical Medicine. Pretty cool name. First author here is Johanna Steffen. In this study, they created a West Nile virus encephalitis model using human cerebral organoids, which we've talked about quite a bit here on the show. This was generated exclusively with male iPSCs, and perhaps, a limitation there. It's just a few cell lines that they used here, and they can use other cell lines too, I'm sure, in the future. But as a proof of concept, it's all right. And what they saw is after infection with the West Nile virus, the purified lysate of West Nile virus, it resulted in uh, heterogeneous kinetics of infection, is what they called it, with an early strong replication window that was potentially leading to viral clearance. And there was a subsequent late peak associated with a more long term chronic infection. Now, they saw, after taking a deeper dive into the organoids, different viral foci in the cortical like areas that are rich in neurons and astrocytes, but void of the microglia. So perhaps it's more of a neuron astrocyte centric mechanism here. And then, as you might expect, they saw some pro-inflammatory cytokines like IL-6, TNF-alpha, chemokines, CCL17, etc., different biomarkers of like IL-1R that were subsequently released after infection and upregulated. So that's kind of straightforward there, right? You would expect to see some sort of innate immune response, pro-inflammatory response in response to infection. So it's a model establishing a model for future studies, perhaps studies looking at antiviral compound development, and just showing that, yeah, you can use these human cerebral organoids, just like which was the case for Zika back in the day, to look at West Nile virus encephalitis. This is a useful model, potentially valuable properties in the model to study not just acute infection, but also potentially a long-term chronic infection. And certainly these models, as we've discussed for years now on the show, hard to believe, years, we've been talking about this kind of stuff. They're very useful. So you name your pathogen, you name your virus, and perhaps organoids can be the way to unravel the mechanisms of that infection.
Yes, infectious disease. I can recall the good old days. I'm nostalgic and longing for the good old days when the panic was about West Nile virus, which now seems comparatively relatively mild. When you think of Zika, which had a real dramatic phenotype, and then COVID, now I'm just numb. Like I just assume that I'm gonna succumb at some point before my time to some kind of infectious disease, whether it be airborne or mosquito-borne. But here I love, and like you said, we've talked about a lot now. I would say starting with this was the Zika was kind of the beginning for the show, I guess, of looking at infectious disease using these stem cell models. And we've talked about it a lot, but this to me had a really fascinating twist because I think over time, one of the major challenges, I guess, that some people are trying to address is that in these organoid systems, of course, depending on how circumscribed they are vis-a-vis the number of cells types that are incorporated, like they lack a immune system, right? This is just a cortical or whatever cerebral they're calling it organoid. Um, so studying infectious disease using a system like that, I can imagine one of the knocks is, okay, that's great on a cell intrinsic, whatever the tropism is, you can look at the effect on that specific cell, but it's in the absence of this secondary influence, namely inflammatory influence from the immune system, right? But here, what was the twist was that in the absence of immune system, they still had viral clearance based on this kind of like cell intrinsic or innate immune response, which I found fascinating. Not totally surprising. I mean, there is this innate quality to glial cells, and cells of origin are immune for glia in part, but still, I thought the system was remarkably, overachieving in that it addressed this innate immune response, and then, of course, there's the inflammatory, secondary response, which is probably underlying a lot of the sequelae of the encephalitis, was very clean, offering a lot of targets that may be addressed by current therapies to the immunomodulatory therapy. So I thought it was really cool that not only they, uh elucidated this kind of neural intrinsic viral response that can clear, but then showed in even in long-term effect infection that there are targets that could you know you could deploy existing or new methods to staunch that encephalitis and subsequent effects. So I thought this was a great study that really overachieved and added a novel angle to the many infectious disease organoid stories that at least that I've seen. So great bit of work here.
Yeah, absolutely. And I think in this day and age, everybody is really obsessed about creating organoid models that harbor that immune component. And in fact, this is something we're going to talk about with our guests today. But there's a lot of utility for studies like these, like you're alluding to, where you're just looking at infectious mechanisms, viral tropism on different tissue types, and the impact of the innate immune system intrinsic to the cells themselves, and potentially clearing out the virus. And I'll take a step back and toot my own horn for a second here. You know, back in the day when I was a grad student in Joe Wu's lab, one of my first studies that I did in grad school was throwing some Coxsackie virus, a viral causative agent for myocarditis, on purified cardiomyocytes. And we didn't have any sort of immune system or anything in that particular model, but it was still useful because you could show that, yeah, you can use this purified cardiomyocyte system as a way to study the infection mechanisms of this potentially severe viral illness of the heart. And then we took it a step further and actually introduced exogenous interferon beta to hyperactivate that innate immune system and actually clear out that viral pathogen, even in these purified two-dimensional cells. So I think there's a lot of utility in these model systems. Things are, of course, a lot more advanced than back when I was doing them, which was just 2D cultures. Now we're talking about organoids. But even stuff like what Xubing Chen, your neighbor, did during COVID, basically looking at a panel of different organoids and seeing the infective tropism for SARS-CoV-2 in different tissue types, there's a lot of utility here. So immune system is definitely hot. It's all about incorporating those immune components into the organoid models, but there's a lot you can do without it too.
For sure. 2D, 3D, organoid, assembloid, they all have their uses and the secrets to reveal. So I'm a fan of all of them, whatever gets you to the answer, right? Speaking of disease, we're actually taking a little bit of a tour of all kinds of disease today with 3 out of 4 of these stories. And then we're going very, very, very basic. But this second leg in that disease odyssey is looking at mitochondrial disease. We're talking about infectious disease. We're going to talk about cancer in a minute. But here we're talking about mitochondrial disease, which, is well described due to dysfunction in cells' energy producing organelles, those mitochondria. And one of the more severe forms of mitochondrial disease that's compatible with life, live born, is Lee syndrome, which is characterized by neurodevelopmental regression and muscle weakness, and ultimately these patients succumb to early death, often from metabolic crisis. And the thing is with Lee syndrome is that it can result from mutations, variants in more than 100 different genes, right? That play a role in either nuclear DNA or mitochondrial DNA expression. And most of those genes, if not all, have a role in oxidative phosphorylation, right? And so there's been many, many that have been described. But a couple of those genes, and we're going to come back to this, are this SIRF1 gene and this NDUFS4 genes that have been associated with Lee syndrome. And of course, maybe not, of course, maybe not everyone knows, but one of the things that's so tough with these is that there's no treatment for mitochondrial, um, not all mitochondrial diseases, and there are methods to try and obviate it with this whole idea of nuclear transfer, which is still, I would say, experimental, non-wide practice. But once these people are born, there's no treatment for Lee syndrome specifically. And what's worse is that there's not a lot of model systems to even investigate and seek out a treatment. We talked about this paper a while back, which was, I think, a biotech paper that was all about mitochondrial DNA editing, which was kind of a major breakthrough because it enabled you to recapitulate in a controlled fashion with isogenic controls and all this mitochondrial disease by doing this focused mitochondrial DNA editing. But still, it's tough, right? There's a lot of challenges to mitochondrial DNA editing. And as such, there's not a lot of animal models that employ this mitochondrial disease targeting. But there are a couple. uh, of major value. There's this pig model that is a knockout of SIRF1. And then there's this, I said we're going to come back to it, that one gene I mentioned before, pig model is SIRF1 knockout. And then the most widely used model of all is this NDUFS4, the other gene I talked about, a knockout, homozygous knockout of that in mice, which recapitulates a lot of aspects similar to the manifestations of Lee syndrome. And the whole idea there is that you could use these to elucidate treatments, right? And there have been some proposed therapeutic interventions, antioxidants, rapamycin, the cure-all, even cannabidiol, right? But only a few of these strategies have actually been deployed in humans with Lee syndrome, and there's not been any report of successful results using these compounds. So this is where, of course, induced pluripotent stem cells come in, right? And these new approach methodologies that we're talking about so much these days. So with Lee syndrome patient-derived induced pluripotent stem cells, you can recapitulate many of the cellular aspects of disease, like neural outgrowth defect, altered calcium homeostasis, glutamate toxicity in these neuronal cultures, and impaired cortical development and neuromorphogenesis in organoids. So, like, again, getting back to it, 2D, 3D organoids, they've all offered some of these candidates that you might be able to target to address Lee syndrome. And the lead of this group, Alessandro Prigioni, who's at the Amalia Children's Hospital in the Netherlands. And this was a group that had many corresponding authors. Alessandro was the lead. There were, like, I think 77 authors on this from more than 50 different, attributions where they come from. So this was a massive group. And you'll understand why when I clue you in on the feast of experimental studies that were incorporated into this one Cell article. It's a big splash, and deservedly so. But anyway, Alessandro Prigioni, almost a decade ago, showed in this Cell stem cell article that you could use these iPS-derived neural precursor cells for as a drug discovery platform for mitochondrial disease. It was at the time, almost 10 years ago, that was huge still. iPS cell and disease modeling, and particularly in mitochondrial disease. And what they found in that study was that there's this score. There's something you can score for, which is key for any kind of high throughput assay, and that's this abnormal mitochondrial membrane potential, right? So using that phenotype in these Lee syndrome specific iPS cells, this massive group, who knows who in the group did each specific assay, but in aggregate, they performed a high throughput screen using these Lee syndrome iPSCs, and hit on phosphodiesterase type 5 PDE5 inhibitors, specifically because of its exhaustively studied safety profile. They settled on sildenafil. Hard for me to even pronounce that because I'm not really familiar with that drug, as I don't use it, Arun. You may also know it as
I
Viagra.
I'm familiar with it, but I also do not use it.
Hey, maybe I do use it. Whatever. No shame in my game, son. But regardless, as I said, this is something that has been very well qualified in the populace, specifically males, but females too. Hey, and they found using this high throughput screen that Viagra, I'm, like, not going to try and say the name, it rescued a lot of the elements of the phenotype in this IPS system, the disease modeling system in the 2D and organoids. Um, and then, and this is why the group was so massive, they went into the NDUFS4 knockout mice. They went into the SIRF1 knockout pigs and showed that they extended the lifespan. So a real, in vivo benefit there. They went deeper, and the mechanism showed that it was mediated through this cyclic GMP-dependent protein kinase 1. And the crown jewel here of this story is they went off-label with 6 patients who had this Lee syndrome with the specific mutation that was rescued in the disease platform. And they showed in these patients, they showed improved muscle strength and resistance to metabolic crises that are often the ultimate cause of death that these patients succumb to. So this, I mean, talk about an arc, right? 10 years ago, dropping this platform saying, hey, we can score for mitochondrial disease and the cell-based platform. And now, after all this time, and clearly contribution of many scientists, and patients and pigs and mice, they've arrived at a therapeutic candidate that is already on the market. Some of these lead patients, maybe having a good time while they're getting better. So I gotta say, this is, and I shouldn't joke because Lee syndrome is it's tragic, and the physical manifestations are really grave. But I mean, it's a great ending to this story in that this is a drug that is like totally safe, and maybe making a difference for a lot of these patients who have nowhere to go. So this is a great story for me, Arun. What's your take?
Yeah, definitely a happy ending to the story.
Oh boy, you said it.
I went there, but but look, man, joking aside, I think this is an example of drug repurposing at its finest, right? Like you said, this drug sildenafil has been around forever. It's been very well studied and is broadly being used. And I mean, the question I have here is the mechanism of action here. So Denafil, Viagra, it's known for its vasodilatory impacts, right? That's its primary function for, erectile dysfunction or whatnot. I mean, is that part of the reason why these folks are improving and these models are improving in their downstream phenotypes? I mean, they literally mentioned this here. They got to study the vasodilatory mechanisms of sildenafil in this particular context because maybe it's the increase in the blood flow and the oxygen delivery to the muscle, the brain tissue that might contribute to the metabolic, beneficial metabolic effects. So it could be a completely off target mechanism here. But it's something that I think they really do have to dive into.
Yeah, I agree with that, and it stuck out to me in the abstract because there's so much science in this story, and so that stuck out to me as like, do you really like they really had to drop in that cyclic GMP dependent kinase thing because I can imagine that was one of the major requests of the reviewers, and it is key. It is key, even though there's a part of me that's like, yeah, there's a lot of drugs that we don't know exactly how they work. There's a huge unmet need here with 0 options that seem to be effective, like, there's a part of me that's, like, kinda like, "Who cares?" But, on both sides, 1, yes, scientifically, the value is key. Also, you know, I'm sure there is a measurable benefit here, and who knows how much that can be extended once now that we have a positive trajectory for these patients, maybe we can optimize and refine and maybe get treatment to earlier patients, to patients earlier and maybe have a wider delta in terms of the benefit. But knowing exactly how the mechanism works, this is why we care about mechanism, is that maybe you could drill down on that specific element of the disease. Like I'm pretty sure the main role of Viagra that we celebrate so much probably has little to do. Maybe the vasodilatory function, but certainly not what's going on below the waist. But the irony there, even was that Viagra, the initial discovery or the initial application of Viagra, no one developed Viagra for that specific function, right? They just found in the patients in clinical trials that were taken for something else, I forget what, were, having some happy endings, so to speak. So I think that this is an odyssey of a drug that was meant for one thing, ultimately changed the world literally toward another effect, and now has swung back to something that is much more clinically focused rather than recreational. I mean, what a cool arc.
Yeah, shout out to these authors and shout out to you, Sildenafil, for changing the world in more ways than one. Moving back to organoids for our third story here, I'm going to talk about a cell stem cell paper titled Paired Patient Derived Organoids Reveal Transcription Factor Driven Epigenetic Remodeling in Breast Cancer Metastasis. So, like you alluded to, we're really jumping around here in the roundup, covering all different types of diseases, model systems. This is a study coming from Fei Xavier Chen whose lab is over in Fudan University, I believe. Actually, apologies. This is State Key Laboratory of RNA Innovation in the Chinese Academy of Sciences in Shanghai. So, breast cancer has a lot of heterogeneity. This is a big problem for its treatment, right? There's triple negative breast cancer. There's all different types of breast cancer. There's no one size fits all. So, this clinical heterogeneity is potentially associated with epigenetic reprogramming during the progression of the tumor. And until recently, there haven't been a lot of ways to evaluate this. I mean, epigenetics has really taken cancer biology by storm, especially in the last 10 years or so. So they wanted to basically do a deep epigenetic profiling of tumor samples, breast cancer tumor samples from a bunch of different individuals, so they could capture molecular changes associated with metastasis of the cancer in the different individuals. So we're really talking about personalized application, personalized medicine here. So they created a biobank of patient-derived primary organoids from matched primary tumors, adjacent normal tissues from these same individuals, and also lymph node metastases. This is a really, I think, cool study, not just because they're making organoids from the patient's um, own cancer tissues, but they're making organoids and, generating the appropriate controls from the adjacent cell types as well. And then they did, deep omics analyses, genomic, transcriptomic, epigenetic analysis of these tissues, and then showed that these PDOs, the patient-derived organoids, can actually preserve tumor-specific molecular signatures, recapitulate epigenetic remodeling that is actually occurring during the disease evolution. So these things are really good models for the progression of the epigenetic evolution of those particular cancers. So the profiling also defined four distinct clusters that were characterized by unique transcription factor networks, pathway activities, therapeutic vulnerabilities that were not fully represented by the traditional classifications independent of the epigenetic analyses. They looked at the lymph node metastasis cluster that was actually predominantly comprising of the metastatic PDOs, which had a bunch of chromatin remodeling driven by metastasis-enriched transcription factors. And when those transcription factors were depleted, it actually impaired the spontaneous metastasis in vivo. So that actually, I think, alludes to a target, potential transcription factor target for metastasis here. I like how they wrapped up the paper in that way. Yeah, it was a deep omics dive, but ultimately led to a node, something that I have alluded to in the past, that could be druggable. So that was really cool. And ultimately, I think it establishes this PDO model system as a way to do epigenetic characterization, potentially in high throughput for different types of subclasses of breast cancer, and also just as a really great mechanistic platform for drug discovery as well. I think it's a really powerful system here, and I think the other thing is this same approach could be extended to other cancer types, right? Breast cancer is not homogeneous. Pancreatic cancer is not homogeneous. Same with glioblastoma, and perhaps you can use this as a conduit to generate other primary patient-derived organoid types as well.
Yeah, this is a kind of a template, as you say, that could be applied to other cancers. I think, yeah, what hit me here is just the volume of patient samples here that they had and incorporated into the study. Very impressive, ton of work. Um, and yeah, providing that very careful, granular evolution of the tumor cells relative to normal tissue, I think is the key takeaway. Obviously, from here, uh, from the story, and the showing that these things are relevant in the transplant model, looking in the mice is a nice crown at the end. But we always talk about druggable. I'm sure, I don't know how, maybe they use the drug to deplete those or to perturb the transcription factor levels. Um, but even if it was a drug, you wonder about how these things can be deployed in a human system. I don't know that this is so much about therapy, although the implications to therapy and our understanding of cancer are clear. I think that having that therapeutic in the angle at the end is what you need to get it into a high profile journal like Cell Stem Cell. But for me, it was more just about the evolution of the cancer. And, just because we've been talking about it, I want to bring it back to the many different types of stem cell based models here, cancer stem cell, right? So kind of different from what we were talking about in terms of pluripotent stem cells, but still so valuable. The one thing that I think is also lacking here, which the authors acknowledge, is that we're looking at this outside the context of the immune system, which in this case is pretty critical. We're talking about lymph node metastases. So this is clearly an interplay with the immune system here. And while this study stands alone, I feel like there's a next stage of cancer organoids that it does incorporate in a more comprehensive way, the immune component. And we've talked about we're going to talk about it with our guests today, about these organoids that incorporate immune cells. But I can't recall many that do it in the context of cancer. And when you consider metastases, I think the immune component is um, very important. So I love it as a standalone, and I can't wait to see this template applied to different questions and problems.
Yeah, I like that summary because I think it kind of lays the next step for the story really nicely and kind of obviously, like you alluded to, incorporation of the immune components, but also the stromal components. And without that, they're not able to take this model to its full potential. I totally agree with you. And maybe you can combine these PDOs with engineered immune cells, just literally take the cells from the same patient, put them back into the organoid, or have some sort of co-culture model, even assembloid model, organ chip model. There's a lot of ways you could do co-culture these days. So excited to see where they take this.
Myself as well. Can't wait. Switching gears. I warned you, we're going back. I don't want to say back. No value placed on organisms here. We're all of this world. But this is a story about definitely a smaller organism than human, and a totally different class of organism. But before I get there, let's start at what we all share. All of us animals, we start from a single cell, the zygote, and that zygote is totipotent. It makes all of our cells. Pretty obvious stuff there, but there's such a spectrum of potency that has been very rigorously examined in the mammalian systems that we typically use those models. But the processes by which you go from that totipotent zygote to the various cell fates that are specified during embryogenesis, that can vary greatly, even within mammals, but like much more greatly across the animal kingdom. And that can be encapsulated in two specific types of cleavage program. There's what's called a invariant cleavage program, where the early stage blastomeres, they are committed to specific fates, and this is typical of a lot of worms like nematodes, also tunkids, spiralians. Are those worms? I don't even know. I'm a mammalian centric. But then there's also this variant cleavage program that we're more familiar with, and the key difference there is that restriction of cell fate and potential occurs later in development, after those early cleavage phases. And that's why you see all the 2C like, 4C like, there's all this different cell stages that have been identified and rigorously examined with respect to their potency. But generally speaking, the invariant cleavage programs versus the variant cleavage programs, they have different levels of plasticity, right? Or specifically speaking, in terms of like an embryo's ability to adapt to perturbation, the invariant cleavage programs are less robust, right? You blow up a single blastomere at a relatively early cleavage phase, and that embryo is not going to make it. Whereas you can ablate multiple blastomeres in early mammalian embryos, and you'll get a full live-born animal. So those are the rules, hard and fast. But there are subtleties, and there's so much variability across I mean, who knows what the number is of different species out there, that we really have a very superficial understanding of what goes on there. So this is meant to be addressed by Mansi Srivastava, who this jumped out at me, this story, which is in Current Biology. It was a great story. Love current biology, although we don't cover it a lot because it's more based in these like animal model systems, more basic. But I remember this story. This is Mansi Sarasas from Harvard. I remember her talking about this at the ISSCR 2025, and I loved that they incorporated this basic science into the program then. And I want to highlight it now because we're so fixated on these systems that I think a lot of basic biology and the lessons therefrom are lost, right? So this is a story about the acele Hofstenia miamia. Um, and what's key about this acele worm is that it has this invariant cleavage program. But as Mansi and first author Amber Ruck described, they have this unexpectedly high potency in their early development, right? And specifically, these embryos, they have this invariant program where in going from 2 to 4 cell stage, they form 2 different cell types, these macromeres and micromeres, 2 of each, which, like the name, the macromeres are big, and those are totipotent typically. It's known, if you break those up early at the 2 cell stage, you can get a full worm. Here they showed that if you get a single macromere at the 4 cell stage, can form a whole organism. And what I also loved about this is they developed this totally new approach using photoconversion-based lineage tracing, which I think there should be more of this in these basic systems. We have the tools now that we can ask these questions that are highly sophisticated in relatively underappreciated animals with a short generation time. And that's what they did and showed that there's tissues that are typically not produced by macromeres during normal development, but they're recovered when you do this recombination. They also showed that all at 8 cell stage, all of the blastomeres, the micro, the macro, they could all be reprogrammed amongst each other and delineate the various fates. So, bottom line here, while I don't know about the clinical relevance, I don't want to make that argument, although it's there. Um, this is a story just about basic plasticity. And, you want to talk about one of the founder inspirational stories for this, you go back to John Gurdon and stories like that, these recombination experiments and these basic models that ultimately, after many years and decades of foundational work, culminate in something like iPS cells. And I think that the lessons we learned from model systems should be highlighted. This perhaps first and foremost of all.
Yeah, absolutely. Don't never underestimate the power of basic discovery and basic uh, developmental biology and basic biology, like exactly what you alluded to. I was going to refer to that, and this is foundational work and similar foundational work that was done by John Gurdon decades ago. Ultimately, culminated in the discovery of iPS, but similarly, foundational work by Jennifer Doudna and other colleagues in the CRISPR field ultimately led to modern genome editing with CRISPR-Cas9 and all these kind of things, right? So it's a beautiful story, and if you wanna learn more about the work that Mansi Srivastava and her lab are doing, we had her on the show, episode 282, a little while ago, and I remember talking to her about this amazing model system of Stenium myiamia and how I believe in her graduate or postdoctoral training, she had to go to Bermuda to replenish the stocks of the worms, and there's just the whole backstory back there just behind the procurement of this particular model system. So please do take a listen to that episode. And yeah, like what you alluded to, I think there's a few things to follow up on here when it comes to the actual imaging and the lineage tracing. But again, just a beautiful reflection on these non-traditional model systems and especially their power for basic stem cell biology. So I love this story.
Yes, I almost forgot about that. There is such an element with these model systems and others that we talked to, and I see at the ISSCR. It's very like Indiana Jones, like where people have to get out there. It's a very exotic and I'd say glamorous view of science. I wish I could get out and wade and find my model organism and bring them back to the lab. But alas, working with cells and NAMs. We're going to talk about all that Well, maybe not the basic model systems, but perhaps are certainly these more sophisticated NAMs and assembloids with our guest today. Before we get there, though, I have a brief message from Stem Cell Technologies. Are you interested in generating cells derived from human pluripotent stem cells? You can get a free wall chart on the directed differentiation of HPSCs. Learn more. Visit stemcell.com/HPSC-differentiation-wall chart. Get out there and refine your diffs, people. All right, everybody. With us today on the show, we have a special guest from Michigan State University, where he is Associate Professor and Director of the Stem Cell Core, Dr. Aitor Aguirre. The Aguirre Laboratory is focused on stem cell and tissue engineering approaches to improving human cardiovascular health. They're developing innovative technologies and models for engineering and repairing human hearts, employing integrated approaches that include the use of pluripotent stem cells, heart organoids, in vivo cellular reprogramming, and tissue engineering. A lot going on there. Aitor, thank you so much for joining us on the show today.
Thank you. Thank you for having me today. Thank you for the invitation.
Yeah, we've loved covering your work recently, and obviously, as a fellow IPS cardiomyocyte biologist, I'm a big fan of what you guys do. So, like Dalen said, your lab really focuses on all things related to the human heart development, disease, modeling, all that with pluripotent stem cells, cardiac organoids, which is one of my favorite model systems as a fellow IPS cardiac biologist. And but your lab is working on a bunch of different things. So we'll let you start things off. Why don't you tell us what you guys are working on right now, kind of in your own words?
Yeah, well, so I want to say I'm also a big fan of the work that you guys are doing, and Aaron, I've been interested in putting stem cells in space also for a while, organoids. So I feel very envious that you guys get to do that all the time. Um, so what are we doing in the lab right now? I think my lab started with a couple of different goals a few years ago at MSU, and we were doing regeneration, we were doing organoids, we were doing lipid metabolism, and something that really like exploded was the whole organoid space, right? So I remember we started doing organoids like we didn't know what was going to happen in the start when we started with the heart organoids. The first experiments were very like exploratory, and they were very successful. And then things took off from there. And now I think 90% of what we are doing in the lab relates to the heart organoids, right? And it's an area that is becoming super exciting, I have to say, from the publications that I see from many different labs all over the world, right? Um, I think things that we are doing right now that we are getting very excited about, um, most of them float around the organoids, so usually the heart organoids. I would say we continue very interested in development, right? Modeling aspects of development. Uh, I would say trying to improve how we mimic development with bioengineering methods, so in vitro, right? Because I mean, it's pure biomimicry. It's not a new idea. It's not an idea that we have invented by any means, right? But I think if we can improve that biomimicry, the organisms are going to get better, right? And so we are working a lot on that, which is, I would say, is technology development, right? So I think our lab does two things very well. One is technology development. We develop new technologies for the organoids, and the other one is disease modeling, right? We try to push new disease models and see who likes them, because sometimes people hate them and sometimes they love them. So, you just throw models out there and we try to improve them, make them as good as possible, and see how the community reacts. But yeah, an area that I'm very interested in is development. Obviously, the other area we are very interested is, maturation technologies, try to make organoids that are more adult relevant, right? I'm bringing in cell types that are important for the heart, but that are not necessarily developed in the heart during development, right? And I'm sure you guys are aware of this because of the recent paper on the heart macrophage organoids.
Mm.
So, trying to bring in immune system, trying to bring in neural crest, derived tissues, and things like that. Those are, I think, the main areas of interest for us right now, and applications of that, basically.
Yeah, I mean, you said you started with this idea that, exploratory, and now it's taken up the lion's share of your research focus, and that's how it goes in science, right? You go where it takes you, you
Yep.
go where the answers and the questions lie. Um, but I want to drill down a little bit more on this. I don't want to call it a dichotomy, because I think there is a through line here between kind of bioengineering and development. And hear me out here, right? Because we talk about on the show oftentimes when we have bioengineers on the show, which it feels like every time we have someone with the bioengineering focus on the show, we have so much to catch up with and catch up on in the field. And some may argue we don't have you guys on enough, but I would say just the pace of advance and innovation is really what's driving that gap. But now that we have you here, I kind of want to start with this big picture view, right? So you alluded to it. There's these model systems we're getting more and more faithful, right? And the buzzword of the last few years, I would argue, has been self-organization, right? And maybe that's two words. But there's these emerging models, not just the gastrula and the human embryo or whatever embryo models, um, but across the board, I think there's these emerging models that are more and more faithfully recapitulating the native developmental architecture of human tissues, which is the most exciting. But of course, again, across the board, all different types of organoids, you name it. But traditionally, and you're gonna talk me out of this, but I think maybe other people share this view, that there's this connotation with engineering of something that is built, right? Which may be seen kind of opposed to something that is grown, right, organically. So can you kind of, because you already kind of alluded to it, that's your focus, is getting biomimicry, right? But you're kind of have to incorporate these engineering principles to create an environment that will enable more faithful self-organization. So can you kind of like tie those together with how do you go about that? How do you engineer a system that's grown? You know what I mean? It seems like a bit of a dichotomy.
I mean, it's a great question, Daylon. I love it. It's also a very hard question to put into a few words, right? So we could be talking about this probably for 1 hour, and we might still have things to say. Um, I think when I say in our lab, when we talk about engineering, I think a large part of it is using an engineering philosophy, an engineering mindset applied to biology, right? Um, when some people talk about tissue engineering or bioengineering, they are talking more about technologies, right? So about building up tissues. I like to put this example because I'm in Michigan, right? Michigan used to be the automotive capital of the world, so I like to put the cars examples, right? Um, there are a lot of people with very solid engineering mindsets, and they want to build human tissues like you build a car, right? So you take the different parts of a car, and if you put them together and you know how to put them together, you make a car, right? And I think that works great for human constructs, right? It's like a car because you know what every piece of a car does, and you know how it interacts with every other part. So you know the parts, you have them, you put them together, you get a car. I think the problem with biology is that we don't know what a lot of the parts do, and we don't know how they talk to each other because we have incomplete knowledge.
Mm.
And so I think that's where self-organization is useful. I think self-organization is useful because we still do not understand biological tissues 100%, right? I think as long as we don't have um, a very, very deep, clear understanding on the different biological parts, right, to use engineering terms, self-organization is going to have a space here. It's gonna be interesting, right? Because with self-organization, what you are doing is um, you are passing some of the responsibility to make that tissue to the cells themselves, right? And you know that they know how to make it. The problem is that we don't know all the communication, all the molecules, and so you just kind of like you delegate some of your authority down there to the cells, and they actually do a pretty good job, but it's not perfect because again we are lacking information. Now, if you think some of the engineered heart tissues in the past, how they have been made, especially when I was a grad student, I remember, because this has been a field that has been around for a while. Um, and people were taking cardiomyocytes from rats, and then hubex, and then fibroblasts, and then they would just put it and assembling everything together. Um, the degree of control that you can exert in those engineered heart tissues is not great either, right? Because again, it's there's a lack of knowledge, and at that time there was also a lack of human sources of cells. Like that's something that we have solved now with iPSCs to a certain extent. Um, and so, I think both approaches, both the pure like, engineering approach, I call it the bottom up approach, right, versus more of a top down approach. Um, it's very valid. I mean, engineered heart tissues are very valid. I think in an ideal world, right, like let's say 60 years from now, let's say we have learned everything there is to learn about how the heart forms. Then engineered heart tissues are going to be superior to self-organizing technologies, because you are going to get to the same result with better control, right? Like what you are handing over to the cells in self-organization is control over what happens, right? That's the scary part. But for the time being, I think in the time we live now, I think both approaches are very interesting and are very useful. So self-organization can do things that we cannot do with engineered heart tissues in vitro right now, right? Like our organisms, for example, organize into chambers with atrial and ventricular myocytes, develop pseudoatrial nodes, develop very sophisticated anatomical and morphological structures of the heart that if you try to make them using an engineered heart approach, I don't think you can right now. Correct me if I'm wrong. I don't know anyone that it's actually doing that level of sophistication yet. So I think both approaches are very valuable and are very useful. Obviously, in engineered heart tissues, you exert more control, right? So that is definitely an advantage that they have over organoids now. One of the disadvantages of organoids right now, I think, at least what we are observing, is that aside from delegating control, obviously, to some extent, is that the instrumentation for organoids is not great. Like microscopes or electrophysiology devices, things like that, they haven't really been designed to work with relatively large self-organizing organoids. Like our organoids are like 1 to 2 millimeters in diameter. So they are, pretty big for organoids. If you want to do electrophysiology, there's really like no instruments designed with organoids in mind. If you want to do microscopy, they are very large. You need to use specialized, instruments, sometimes like in-house developed instruments. So, I think that's one of the disadvantages of the organoids too, versus engineered heart tissues, where you can have like posts that contract, and you can measure force, and you can measure electrical activity. Like people have been designing those for many years now. So this is kind of like pre-existing technology there to better interrogate those models. And I think organoids are still lagging behind in that respect.
Yeah, it's a really good point, and it's something that my lab's, looking at as well. And I think there's specialized pieces of equipment that are starting to emerge for organoid
Yep.
imaging, and even like you've probably seen some of these basket MEA approaches for field
Yep.
potential
Yep.
measurements for cardiac and cortical organoids as well. But there are a dime a dozen. There's not too many out there, and you're right. The engineered heart tissue field, I think, has had a head start. And we're thinking about like what you're doing, making these organoids more advanced, creating assembloids, advanced co-cultures with immune system, immune cells. I mean, it's something that you guys have done so well, and we've highlighted your recent cell stem cell paper on the topic of actually developing this human hard macrophage assembloids that can mimic immune cardiac interactions, and it's a very very hot topic, and you guys have used this to enable advanced, arrhythmia disease modeling, for example.
Yep.
So we've talked about assembloids a lot on the show, often with like Sergio Pasca, for example, on the cortical and brain side of things. But I mean, cardiac is certainly starting to take off as well. So tell us more about this work, this immune co-culture work, and what's the major application and the next steps there.
Yeah, so this work started quite a few years ago. I think like around 2020, we started, we started thinking about this idea, right? About introducing immune cell populations. And it seemed like, in the 2010s, I read this paper by the Holtzman's lab that works in the mouse, and they work in macrophages. And they published this paper in Cell where they show that cardiac tissue resident macrophages in the developing mouse heart were found associated with the conductance system of the developing mouse heart. And I thought that was amazing because I never thought of immune cells doing things like that, right? I really love that paper, and the data was also so beautiful. So, that's kind of those things that I'm sure this happens to you guys too. Like it keeps nagging you, like over the years, you see another paper and you get back to that one and you think about it, and it just kind of floating in your head. And so in 2020, I got a student, a dual degree student, actually, a DO PhD student. MSU has this doctor of osteopathy school that it's very strong. So we have DO PhDs and MD PhDs. Which is called Colin O'Hearn. I want to give him credit for his work because he has done a fantastic work here. And so Colin came to my lab and I was talking with him about, cardiac tissue resident macrophages, and if we could perhaps, model how they integrate in the heart and try to model aspects of development with them. And so he started working on this, and it took a lot of effort. I mean, I'm not gonna lie. Like there were times that Colin and I were just like pulling our hairs out of our heads because there were so many things that could go wrong with this. Anyway, it was a journey. We learned a lot, but eventually Colin got it. He got these macrophages, integrated in the heart, and then he did extensive single cell sequencing, and we had all the markers. And a lot of the stuff that has helped us benchmark the organoids too is all the people doing human single cell RNA-seq atlases, right? Multiple papers now that have come over the last couple of years, like single cell RNA-seq datasets in developing human hearts, which, it's complicated to do too. And so we used those as benchmarks, and we could see that finally Colin got to have those macrophages in the heart with the right markers, the right integration. Then he tested functionality. They seem to do the things that those macrophages are supposed to do. And one of the things that we saw is that they were interacting with the electrophysiology of the heart. They were connected to the syncytium. The electric signal was going through them. They express ion channels. So, it kind of was confirming the observations in mouse from the Holtzman paper. So very, very excited, right? And then at some point, it was like, oh, this is super cool, Colin, but when we submit this for review, I'm sure the journal is going to ask, what's the biological relevance, right? Like, how do you apply this to sub-disease model? It's always the same scheme, right? And so we were thinking hard conditions to apply this to, right? Like a disease model we could introduce where this was a relevant finding. And, Colin was exploring different areas, and one day he said, oh, I want to put these pro-inflammatory cytokines and see if it does something, and see if we can model like atrial fibrillation, because this is a very prevalent condition, and it would be useful. And I'm gonna be totally honest with you, I was very skeptic. And you guys are laughing a little bit, but yeah, I was extremely skeptic at the time. But I said, okay, let's go ahead. I mean, we have nothing to lose, and this might work. And it kind of worked, right? So I was super excited when Colin showed me the results. And then part of this also came through a clinical collaborator, Naguib Shalfoun at Corewell Health, which is a hospital system here in Michigan. And he's an cardiologist and electrophysiologist. He works a lot with atrial fibrillation patients. So his feedback to fine-tune this model was instrumental, right? Because he gave us a lot of information about phenotypes that we wanted to look for, and the relevance in patients, and like a lot of conversation and discussions. And yeah, that's a little bit the story behind that paper, right? And the next step on this is we would like to set up a high content screening platform. so that we can start screening drugs for atrial fibrillation, right? Investigate perhaps anti-inflammatories or some types of cells that will mitigate that cardiac tissue resident macrophage dysfunction that we see in these organoids under atrial fibrillation conditions. I have to say it's challenging because there's not a lot of agencies that like to fund, screenings and platform development and things like that. It's kind of challenging to push forward, but we are very dedicated to this, so we are going to continue trying. Yeah.
Yeah, well, I mean, what they should think twice about it, right? Because these models are so powerful and disease relevant, as you've established, not only because they're human, but because you find you make these observations that are like direct lines to the clinical pathology in a considerably unmet need. But the best part for me of that story is to take away, a couple things. One is kind of a callback to what you started talking about in terms of having incomplete information, and that's the value of like letting the system speak for itself, right? Is that who knew that these non-cardiac cell immune cells would have such a role, even though the observations may be made in a different animal model, recapitulating in a human embryonic developmental system. It's so beautiful. And then really the best part of that is that your skepticism, it really is a lesson that we need these fresh minds to try things that we may not think are the best utilization of time or that we wouldn't commit to, that end up being a, home run swing. So there's so much in that story. And, part of that there is Colin's role. I got a kind of related note about Alexandra Costina, who was on that paper when it came out in CSC. But this is about the bioarchive, because I remember that paper first dropped on bioarchive and then ultimately was published in Cell Stem Cell. And I'm actually interested in this other paper that you kind of alluded to earlier about NeuroCrest. That is, currently still on the bioarchive and presumably in peer review from Alexandra. And I'm going to come back to that in a minute. But first, now that we're talking about the bioarchive, because you seem to be, early adopter there, even though it's well established nowadays, and preprints are like a critical part of science dissemination writ large. But having had that experience and seeing both sides of it pre-post publication, having something currently on the archive and in review, what's your advice to anyone considering posting, specifically with regard to like when is the story complete or mature enough in your view that you put it out there? And also, what's like the biggest value add for you as the scientist posting that on the bioarchive? Like, there's a lot of reasons to do it, but for you, what have you noted has been the biggest return on that?
Wow. Yeah, that's a really good question. I think, you know, I think there are advantages and disadvantages of putting things early in bioarchive. So, well, like early between quotes, I would say here, we have kind of like, I have my own internal rules about when something can go into bioarchive, right? Which is when we basically consider that the story is sufficiently, rounded up that it's ready to go for peer review, right? So whenever we put something in bioarchive, we immediately say to peer review. But we want to give the scientific community an early taste, let's say, of what's going on. I am a true believer in open science and making science available to everyone as much as possible. And bioarchive does that, right? So anybody can download it, anybody can read it. Um, and so I think bioarchive kind of like it's hit and miss sometimes because you can upload a paper without methods and things like that, or with very complete methods, or some people use bioarchive to just basically put full preprints with all the methodologies, everything, right? And some people, they just use it as, I don't know, like a half attempt, but they don't put enough methods that you can reproduce, so it's just not like kind of complete. I mean, I don't have strong opinions about it, but I like it to be, yeah, I kind of like it to be at the first, take at our publication, right? So I treat it as a publication. Um, And then we move on from there. Costina's paper, right, Alexandra's, it's been in peer review since then. It's been rolling around, and it's complicated because Alexandra also had a baby in between. So congrats to Alexandra. And so she took medical leave, etc. So that has delayed things a little bit. I think we'll see it out soon at some point. But it's a very exciting paper. It's getting a lot of citations, even as a bioarchive in big journals, like in Nature review journals and things like that. So kind of happy about it. But yeah, I would like to see it finally published at some point.
Well, it's a benefit to us all, and for my own interest, I mean, I was drawn in by that study because of the neural crest angle. My own work has an angle related to the adrenal gland, which I just stumbled into. Like I said earlier, you go where the science takes you. The medulla, maybe for those who don't know, I'm sure you know, the medulla of the adrenal gland is derived from these neural crest derived chromaffin cells, right? And the adrenal medulla is responsible for dumping adrenaline into our bloodstream. So emblematic, I would argue, of this promiscuous purview of neural crest cells. They go all over and become a lot of things, right? Across development. So can you elaborate a bit on this for our listeners here? How does the incorporation of neural crests into these cardiac assembloids augment the model? And how do you envision applying that moving forward?
Yeah. So the whole neural crest story started kind of similar to the macrophage story, right? So if you see a developmental plot of how the heart forms over time, you will see that at some point embryonic macrophages migrate, and at some point neural crest cells migrate to cardiac neural crest cells to kind of like put all the fine details in the heart, right? Like finalize the outflow tract, the septum, innervation. They do a lot of things there. And so the idea was like, we are trying to build as sophisticated heart as we can, right? And so, again, developing a protocol to do that was kind of painful, even though there are good neural crest protocols out there. But we finally, the integration was perhaps the most delicate part. We came up with an approach where we create kind of like neurospheres that are placed adjacent to the organoid, because we don't want to force the neural crest cells to go inside. We want them to actually migrate inside, right? And I invite anyone that wants to know more about the details to just check the bioarchive, because it's complicated. It's a long story. But regardless, the neural crest cells are really interesting, right? So they developed into several different mesenchymal populations in there, similar to the outflow tract cells. Problem in the organoids is that they don't have enough anatomy to actually be able to say this is outflow tract, right? It doesn't develop correctly. This is just like a mass of cells with outflow tract like markers. But I don't dare to call it outflow tract because, I'm sure developmental biologists would come and, shake me up. And for good reason too. I'm not complaining. So, but they do have this profile. But they do many other things, right? And one of the things that we observed very quickly was that there was a lot of, elongation and, processes looking like axons and dendrites and stuff like that. And we were like thinking, oh, perhaps they are actually developing into elements of the conductance and innervation system in the heart. And yes, that's the case. It turns out that the parasympathetic innervation in the heart derives from the cardiac neural crest, while the sympathetic innervation comes from the trunk neural crest cells. And so we are missing that part, but we only see the parasympathetic innervation. And this parasympathetic differentiation, actually, the cells innervate all the way down to the sinoatrial nodes that we have in the organ. It's actually pretty cool, and we can see the connections to the cardiomyocytes there, like the axons end on top of the cardiomyocytes and all that. And so it's a very interesting model to observe how actually innervation develops in the heart, right? So we've been trying to characterize those neuromuscular junctions that form there and all that. And then we observe that, these organoids beat slower because they have parasympathetic tone, and then we can modulate them by modulating the activity of these neurons. So if we can add like stimulating, like neurotransmitters or drugs that are related to them, then we can regulate the organoid bit. The motivation, I think one of the big motivations, or at least one of the areas that makes me excited about this, is that it opens a whole area where you can start to study eventually in the future the brain heart axis, right? Like the sympathetic, the autonomous nervous system connection to the heart, and perhaps someone that can make, like brain organoids from the relevant parts that connect to the heart, which I don't really know right now what it is. Um, you know, create some kind of assembloid that has that brain component, the autonomic innervation, and the heart component to study sympathetic and parasympathetic tone in the heart, which are important for heart function. And for example, in heart failure, there is this increased sympathetic tone right on the heart, this neurohumoral component. Um, I've got a lot of people calling me about this paper that study Parkinson's and Alzheimer's and things like that, and they tell me, that there's this implication of Parkinson's in the heart, and I'm like, no, I didn't know that. So we are very interested in your system because perhaps we can study how it affects the heart. I'm like, I'm all for it. I don't know I don't know how I can help you, but I'll do what I can. Um, and I like this science because it's very exciting, because sometimes this is following, this takes me out of my zone of comfort too, right? Like I don't know anything about brain organoids, but I'd be happy to work with someone or start exploring these areas more, and learn in the process, right? So I think it's really exciting science. I don't know where it's going to take us, but I think it's very exciting.
Hey, I mean, that's the best way to do science is just be excited by it and see, where the results take you, right? I mean, as somebody who does a lot of collaborations with neuroscientists myself, I can appreciate the enthusiasm there. It's pretty cool, and a lot of the tools, like we were talking about for electrophysiological analysis, all that, there's overlap there, natural overlap with
Yeah, yeah.
cardiac and neural as well. So it kind of works out in that way. So before we let you go, we wanted to shift gears a little bit, talk a little bit about where you're at. You're at Michigan State, perhaps not traditionally known as a powerhouse in stem cell biology, but thanks to your work and the work of others, has really emerged as a leader in the field. Just as a side note, your new chair in the Department of Medicine is somebody who I've known for some time, Dr. Chaz Hong, and he's a cardiologist, cardiac biologist himself. Known for his work on PI3 kinase, BNP signaling, and more recently in pluripotent stem cell cardiac modeling as well. So, tell us about that environment that's facilitated all the amazing work that you're doing right now in your lab. So, tell us about Michigan State and what do you love about being there?
Yeah. So, I trained at San Diego in California, right? At the Salk Institute and University of California, San Diego. And, I love it there, by the way. I love San Diego. And then, I was contacted by MSU, Michigan State, because they were making like big recruitment efforts. There was this new initiative called the Global Impact Initiative. And they wanted to bring like, high impact researchers to Michigan State and develop a lot of the biomedical area, right? So as you said, MSU has traditionally not been known by its biomedical research, but that has changed a lot in the last 5 to 10 years, and it's accelerating. The rhythm of change is accelerating. MSU just fused with the Henry Ford Health System here in Michigan, which is the largest medical provider in Michigan. MSU is very uniquely positioned too in some respects, again, because it has a medical school, like a traditional medical school, but it also has the College of Osteopathy, which is kind of unique. And, we just got the first T32 in the country for the osteopathy program. So, it's a lot of new stuff is happening, right? We are trying to attract talent in the stem cell field. Like you said, Chas Hong is here with us now. Um, and what I can tell is that there's a lot of interest. There's a lot of growth, a lot of investment in this area, right? New buildings that are being filled up with researchers in stem cells and other topics. And so it's a very good place to start new things because there's opportunity. And the cost of living is cheaper than in California, so that's one of the selling points. So yeah, that's a little bit the situation here.
Yes, on the rise for sure, and in large part, I think due to, the models that you've introduced, and the collaborations that you've established. So appreciate that. I'm sure MSU appreciates it too. Before we let you go, we got a couple of closing thoughts. Kind of pick your non-scientist or science peripheral personality and brain. First, if you could answer any scientific question, what would that be?
Well, there's many scientific questions that I would like to answer. Which one is the top? I would say, I have a big interest, like a hobby outside of not outside of science, but outside of what I do in the lab. I have a big hobby in astronomy. I love astronomy, and I have a telescope. I take pictures of the stars, and and everything is automated. I have like computers, and it's a big investment, believe me. Um, the James Webb Telescope, right? So this is something that fascinates me, like the stuff that NASA is doing with the James Webb, and the things we are learning about the universe with the James Webb. And so one of the questions that I would like answer perhaps one day, right? It's about all of these things about the origin of the universe, right? Like we have the Big Bang, and we thought that the universe was like 14 billion years old, but now the James Webb says that it's seeing things that are beyond that margin, right? So like the universe is actually older, and so I don't know. I would like to know more about the origins of the universe. Perhaps what are we really doing here? I think that's the basic question. Where does everything come from, right? Like that's something that, and I mean, it's scientific, but it's also kind of philosophical to some extent. But it's something that really, I think about it a lot. I mean, it's not that it's very useful because I'm not going to find the answer,
Yeah.
but really, that's one of the things that gets me really excited.
Yeah, that is something I try not to think about. It's one of those ideas that's just so big, like I fall into delirium, the scales of time and space there are just so great. It's almost like inner space when you go subparticle, I'm
Yeah.
very comfortable in the nano to micrometer range, I would say, as a scientist, and that's where it stops. But hey, you go for it, my man, and when you get those answers, let me know. So yeah, we got a little bonus question there in terms of your hobby, astronomy, which would be, I think, cool. Do you ever take pictures? Could you have a camera hooked up to your telescope?
Yeah, I actually have a very nice CMOS camera that can take fantastic
Oh
pictures. Yeah,
boy,
so
well, if
it's a hobby of mine. It connects to microscopy too, because I do a lot of microscopy. We do a lot of microscopy in my lab, right? And it's almost an art to take good pictures sometimes. And so I like the very small scale and the very large scale, I guess.
No, for sure. I mean, Arun would agree with you. You can't see it behind him with his blurred background, but he's got a picture of the pillars of creation there that I see the same thing. You look at some of these micrographs that are very small and some of these James Webb or whatever telescopic pictures, and the parallels are undeniable. So there is some
Yep.
symmetry in the eyes of the creator, as they say. I'm going a
Yep.
little bit too deep there. Finally, we got three for two here since we got a little bonus there, but just a piece of parting words of advice for trainee scientists at any level, based on your experience in bioengineering on the cusp of developmental biology.
Well, I would say my piece of advice for, like new trainees is if you want to be successful, right? You need to work smart. You need to work hard, but you also need to work smart. Use your time wisely. It's not always just putting hours. I mean, you do need to put hours to a certain extent, but it's also using your time very wisely and don't do unnecessary experiments. Just plan your experiments well ahead. It can save you so much time, but also it's so important to work in a team.
Mm.
The science is changing. It's becoming bigger and bigger teams, bigger and bigger collaborations. The problems we are facing are bigger and bigger. So like the romantic idea that the public has of the lone scientist in his lab making world leading discoveries, I think that's disappearing, right? We are, bigger and bigger teams. And so I always say that my lab is like a soccer team. I'm from Spain, so, soccer is big in Spain. And I always say Spain is not good because we have a Messi. Spain is good because we have a good team, right?
Mm.
And so that's what I aim for my lab. I try to have a very, very good team that integrates very well, where everyone supports each other. So as to try to get the best out of everyone, right? So Perhaps that's my piece of advice for trainees.
That's great advice. Work harder and smarter, and you can't do it alone nowadays. So I think those are all things that I've learned, although I still banging my head against the wall on some of them. But sage advice, Aitor, and a lot of great kernels from this conversation that I think our listeners are going to enjoy. So thanks again for sharing, and thanks for doing what you do.
Thank you. Thank you so much, guys, for the invitation. I really enjoyed chatting with you, and looking forward to connecting again in the future.
All right, that brings us to the end of this episode. Don't forget to subscribe to our newsletter at www.stemcellpodcast.com to get the show notes, including an episode summary and links to all the Interbeam Roundup papers. If you enjoyed the episode, please share it with a friend or a colleague, or put it on social where I won't see it because I don't exist there, but plenty of others will. You can also reach out to us on X at STEMCELL Podcast, Blue Sky at STEMCellPodcast.com, or by email at info@stemcellpodcast.com with feedback or to suggest guests. Until the next episode, you guys, thank you so much for listening. We'll be back at you in a couple weeks.